Hello everyone. Welcome to the Bioinformatics Practical sessions. In this session, you will perform a practical on pairwise alignment of sequences using FASTA algorithm. Here, in the LMS platform, we have the procedure that is shown as the fifth item. You can see that pairwise alignment using FASTA is shown here. So before proceeding with that one, we have a prerequisite. We need a protein sequence in our hand. In our previous experiment, we downloaded a FASTA sequence from UniProt-KB. So this is the protein sequence. You can copy this sequence as such. Okay. Now you open the practical procedure. The aim is to perform a pairwise alignment of sequences using FASTA. Okay. For which you require to open the website ebi.ac.uk tools SSS FASTA. So you can open this link in a new tab. So here is it. Now in the practical session, we will have some steps like you have to enter a query sequence or upload a FASTA file containing the sequence. And also you require to set some parameters for running the program and selecting the metrics. Then you have to submit the query. Finally, you will have to observe the results. So that is the procedure. Here, you should select UniProt-KB bar Swissport manually annotated database section. The TREMBL section is actually a computer native tool, which is not actually a protein that is expressed, but it is a computer translation of the gene sequence. So if you are looking for an actual protein, a sequenced protein, then that will be this database, UniProt-KB or Swissport, the manually annotated section. So you can select this one as the database. Coming down, you have to select which sequence that you want to give as an input. Here, the protein database is selected. Now you require a protein sequence to be pasted here. So in the previous example, we downloaded the pyruvate dehydrogenase in the previous section. So this sequence can be copied and there is segment without the command line. So you note that the second line is, or the actual sequence is copied, not the command line. So you can paste it here. So the sequence is now pasted in the search box. There is also another option in which you can browse the file, or this opened file P53170. This can be located and uploaded. For example, you can download, go to the downloads and locate this one and open. So it will do the same purpose. So either you can paste the sequence or upload the file. Okay. Only one is required, any one of them. Now the next step or the third step is setting the program. Here we have the FASTA program or different FastX, FastY, and there are other variants of the program is there, but you require to run the FASTA program, so select FASTA. Now the last option is click on more options to set the different parameters. So FASTA is selected. The metrics, in the theory sessions you might have learned various types of matrices like Blossom 50, BLASP 62, Blossom 80, Palm 250, Palm 120. So various types of matrices are there. So by default it will suggest Blossom 50. You can keep it as such, and you don't need to require you don't have to change the parameters if it is not intended. Anyway, there is an option for changing the metrics, and even the gap opening penalty and gap extension penalty and the k tuple values, etc., can be changed For example, if you want to add to change the or if you want to add more penalty while inserting a gap, you can change it. Or while extending the gap also, you can increase the penalty value so that the entire score will be created based on that one. So this is the different parameters that you need to set. Now click on submit. If you want to get the result by email, you can check on the notify email box. So the job will be running for a few minutes or so for a few seconds. And if it is a very long sequence, and then it will take some more time. So in that case, if you are selecting the email notification option, you will get the results in the email. So this is it. You can see that by after running the FASTA, you have the results. Here is the pyruvate dehydrogenase sequence. It is having 100% similarity. So for 91, this is the actual sequence that we downloaded. So it is giving 100% similarity to exactly to the same sequence. And so there are other options also there, like 23% similarity to other one. So various other peptides with different similarities were listed here. So the length of the query is 491 actually. So it is 100% matched to the same gene that we downloaded. And this is the score. And the higher the score, that will be better. So it is having 100% identity because it is the same sequence. Now this is a shorter peptide with 71.7 score. That's only 1/10 of the score, and also it is having only 23% of the identity. So you will have various scores here. So except this one, the other peptides were not similar to the query sequence. So now you identified the exact from the row sequence that is given to you. You identified the protein from the database by searching it using the FASTA algorithm. So this is pyruvate dehydrogenase is the protein that we used. So this is it. The same exact sequence is now listed in the database after the query. There are other tabs in the results like tool output, where different proteins with the similarities and their scores were listed here. There is a visual output, similarity alignment of the different database sequences to your query is provided here. Functional predictions. So the protein family domain prediction of various hits that is found. It is also listed here. So this is about FASTA algorithm that is used for a pairwise sequence alignment. So this is used for identifying the protein or unknown protein using a pairwise alignment with the database sequences. So in this case, the database used is the UniProt KB. Our unknown sequence will be searched against the UniProt KB database using FASTA algorithm, and we exactly identified the similar sequence in the database using this search. Okay, thank you.